Hey, Mr. Zimmer, how are you?
I'm good. Hey, everybody.
Hey, thank you so much. Welcome to our AP Biology class. We really appreciate you coming in. I truly think you are probably the best advocate for science literacy that we've got. We've enjoyed your writing for years. You write on a various number of topics, things that cover pretty much everything that we learn about in AP Biology. So the students have gone through, they've read some of your articles from New York Times. We've looked at a few of your videos online, and they prepped up some questions for you. I think we're going to try and start out with kind of some personal interest type things, and then move on to COVID, and then jump into some of your other articles. Does that sound all right?
Sounds great.
All right, awesome. Here we go.
Perfect. So I'll start off with the first question. It's kind of a personal one. So throughout all of your writing, I was really intrigued. I want to know who was your greatest inspiration, and it could be whether as when you were a young kid or even into your adult years. Who kind of inspired you?
I guess I've always been inspired by writers who I really looked up to, and they could be sort of writers from long ago, like Herman Melville, or more recent ones. Certainly, in terms of becoming a journalist and writing about nature and science, people like David Quammen have been, a very important influence to me, just to sort of show how it's done, like show that you can write about these things and it can be as compelling as literature.
Thank you very much.
So the next question is, what was your favorite science class you took, whether in high school or college?
It's interesting. I don't think I really had a very good science education, honestly. I remember, I write about biology all the time, but I just, my biology class when I was in high school was not good. And I think that's just, that may be a generational thing. I don't think a lot of biology classes back then were very good. There was lots of things that were happening in biology, in the, in the 1970s and the 1980s, but, I wasn't really getting access to it. So, there was a physics class I took in college, which, I enjoyed, and I'm glad that I took it. But, sometimes scientists in particular would talk about, a teacher that they had in a class they had, and that was it. Like that, they totally the clouds cleared and they knew this was what they wanted to do. It took me longer to make that realization. Biology classes today, when I talk to high school biology classes like yours, I'm kind of amazed at some of the stuff that you guys can do. There are more opportunities to use to do, learn how to do experiments with some of the tools that biologists themselves are using right now, and to even do some real research, so the stuff is there. It's just a question of how much opportunity do you have to play around with this stuff, to learn, to understand how science works.
And then my next question is, what is your advice for students interested in journalism and media, and what would you recommend for student journalists?
Start writing. Don't stop. Don't wait. Just start now. Writing is just one of those things that takes time to learn how to do. Journalism, if you've never called up a stranger or walked up to someone and start, asking questions of them, that's a skill that, it's, you have to learn to do journalism. And it's when the first time you do it, it's kind of scary. The second time, it's a little less scary. And so, there are, the nice thing about writing and about journalism is there's nobody that you don't have to ask anyone's permission just to write. You do have to but and there are a lot of opportunities, whether it's your student newspaper or local places that might be interested in freelance work. Just start. And high school is not too early to start. College is not too early to start. Some people go to graduate school for journalism, and that can be the right thing for some people, but it's not a substitute for just learning, just trying to make writing a part of your life and seeing if you like it. Maybe you will, maybe you won't. I mean, it's one thing to like the idea of being a journalist. It's another thing to actually like doing journalism. It's one thing to like the idea of calling yourself a writer. It's another thing to like writing, because that's what you do. You just do it every day. So try it out.
Hey, Mr. Zimmer, my question was, what is your process of writing a book from a day-to-day perspective to the year timeline of publishing?
It depends on the day. So, I'm working on my next book, and, I've been working on it pretty seriously since the summer. So now I'm at the stage where I'm working on the manuscript mostly. I still have some reporting to do. So, so tomorrow I have a video, call set up with someone I've been talking to before. I've been gone through my draft and, looked at my notes, and I've tried to tell her part of the story, but I can see that there are a bunch of things I didn't ask and that I don't know. I want to, there's a gap, several gaps in the story, and I just don't have the information anywhere to fill those gaps. So I need to talk to her. So at this stage, I've got, lots of notes from visits with people, out, kind of rough outlines from, I'll read scientific papers. What you see right there, that's a table I had to move in here because I needed a place for all the books for this new book. So those are all just for, my new book. And I hold on a second. And anyway, I have there's some more. It takes a lot of books to write a book, plus a lot of in-person reporting. So that's where I am right now.
Can you tell us what it's going to be about? We get a sneak peek.
I have a few more months before I'll feel comfortable really talking about it too much. I need to tame this beast.
Hello, Mr. Zimmer. My question was, you just said you had all the books in the background. Are there any books you're reading currently, whether it's for work or for pleasure?
Well, I mean, I'm reading all that stuff for work. Yeah. I have been, I actually have been, I have been enjoying, I mean, it's not healthy to sit here all day long. So, I get out, go for a hike, go for a walk, go for a run, I will often, listen, also in the car I've been listening to audiobooks just as a way to, for my own pleasure, but also as a way to sort of enjoy some really good books. So I just read/listened to one called "The Making of the Atomic Bomb," and it's by Richard Rhodes. It came out in the 1990s, I want to say, and it's really the definitive history of how our nuclear age began. And it's beautifully written and has a huge cast of characters, and it's really amazing to me how, you have all these different people from, from Albert Einstein to Harry Truman, all these people you've heard of and lots of people you haven't heard of before. And it starts basically with, the revolution in physics in about 100 years ago, where, physicists realized that, all the stuff that Isaac Newton had taught us about, forces, gravity, the way that things interact, it's all well and good for things our size. But when you get down to the atom, a whole new set of rules apply. And they realize, oh, actually, there's, what's holding these atoms together is actually like a tremendous force. And if you unlock the atoms, you can actually produce a huge amount of energy. And so it goes from that realization to how we ended up with nuclear weapons. So it's a really impressive book.
Any podcasts you're listening to these days?
Oh, yeah, too many podcasts.
All right.
My name is Aditi, and when I was reading your Origins of COVID Pandemic, I was wondering, what do you think is responsible for the COVID-19 virus, the raccoon dog or the lab leak? And do you think we'll ever know the full truth?
The problem, the challenge, I should say, with diseases in general is, nobody is there at the very moment that one begins. Like, well, the people are there who get sick, but they don't know that they're the first people. There's no scientist there, taking notes and documenting everything. So it is impossible to know, with some sort of like perfect accuracy. But, the science does not operate that way. When we try to understand, how the dinosaurs became extinct, like we weren't there. It was happened 65 million years ago. So, there would be, and yet that doesn't mean we can't learn something about it. And so, I mentioned David Quammen. So he wrote a book called Spillover a few years ago, which is, a very masterful look at, emerging diseases and looks at a few particular ones to try to like talk about, well, where they came from. And, in some cases we just don't really know, like with Ebola, we don't really know. I mean, Ebola keeps sort of spilling over every few years. From where? We don't know. We just don't know. Now, just because we don't know doesn't automatically mean like, oh, well, there must be a lab somewhere where it's spilling out of it repeatedly. Like that's not, science doesn't work that way. There are lots of reasons to believe that they are, that bats are spreading them. There's good plausible reasons, because they're relatives of these viruses that are in bats and a lot of other lines of evidence. So it seems like somehow they're getting out of bats and into people and then spreading among people and then disappearing. But that's kind of fuzzy. We actually have a better idea with HIV, even though that spillover probably happened over 100 years ago. And there, what appears that happened is that, people were, hunting for primates for food. Chimpanzees, monkeys, even gorillas. And some of those primates were infected with different versions of the same virus. And in a number of different spillovers, this virus called SIV adapted to people and became what's called HIV. And then it spread from there. Again, nobody was there taking notes, but all the evidence that's been gathered over the years is pointing that way. So, with COVID, like we're still, I mean, I know we all feel like we've been living with this thing forever, but we've only been, the opportunity to study this virus has only existed for 3 years because we didn't know about it before. There it is, from the start, the World Health Organization has been complaining that China has not been, providing more of the data that would help to resolve this. And we're just in the news this, in the past couple of weeks, my colleague Ben Mueller at the New York Times has been covering this, that there was a whole lot of genetic data from the market where a lot of scientists do think that it got its start. And it's only become accessible now. I mean, they literally we're taking swabs off of tables and walls and cages over 3 years ago, and it's only becoming available now. And so that's a real problem. And so, it's going to take a long time to keep working through all this evidence. As there's a lot of debate over, how COVID originated or how it might have originated. I think it is significant that there is no that people who are saying, oh, it must have been in a lab, have no record of this virus or any virus like it actually being in a lab in Wuhan. They're just saying, well, maybe it was. Maybe if we could look at all the notebooks, we could, learn something more about what was going on there in those labs. But you would, that kind of research would still have to explain the evidence that now exists in favor of the idea that animals that were being sold at the Huanan seafood market were the origin. So, for example, why is it that if you look at the sampling, that we know of early cases, why are they all either connected to the market or just in the neighborhood of the market? People who had nothing to do with the market, but who lived around there, they tended to be people who were getting infected first. Why was that? Or, it seems, from, there's an analysis that shows that, in the market, you had the two main branches of COVID already that would end up spreading all over the world. So, why would you find those there? If it just came out of a lab, then you just expect it just to go everywhere from the area around the lab, and that's not what happened. So it's science. This is a process that we will be observing going forward for years. We have some evidence, but there's still, an active debate about what that evidence means.
So I'd like to piggyback on the similar article with regards to if there could be another disease or virus such as COVID to appear again so abruptly and cause as much damage as it did. Do you think that's possible if another one could come up?
I mean, I'm sorry to say, but yeah, people have been warned, this has been something people have been warning about for, I don't know, I guess about 35 years. I mean, I can just point, people have been talking about this thing, this kind of thing happening over and over and over again. And if you read Spillover by David Quammen, he was talking about this sort of thing. And and with coronaviruses, we'd already had 2 coronaviruses, SARS and MERS, that jumped over and caused a lot of trouble. So we know, just if you just look at coronaviruses, you know, SARS and MERS led people to start looking for, you know, coronaviruses in animals, saying like, well, maybe there might be another spillover. And when they look, they're like, whoa, there are a lot of coronaviruses out there that that are in animals, and you know, and some of them are going to have the potential to spill over. So yeah, so we have to be ready for something else, and we can, you know, as rough as this pandemic has been, I mean, there are, it's important not to lose hope. That, I mean, like there are things that can be done. There are definitely things that can be done. There are things that we did do in this pandemic that saved a lot of lives. We need to prepare for those, and there are a lot of things that we knew we should have done that we didn't do, that we need to do better. You know, I'll just give one example. So in the 1990s, President Clinton created something called the Strategic National Stockpile. And this was a whole lot of equipment. They were actually worried about like someone using a virus or anthrax in a terrorism attack, but they just were like, okay, we need to be ready for something sudden happening. So they put in a lot of vaccines into the stockpile, and drugs and masks and things like that. And then over the years, people have, you know, built up that stockpile, particularly to get ready for an influenza pandemic. And, you know, masks, people knew masks are important, especially like in hospitals, like where you're working closely with patients, you know, patients are, you know, giving off viruses. You know, but the problem was, for years, the mask supply went down and nobody was building it back up. And, you know, that was true through several presidencies. And so, you know, at the start of the COVID pandemic, you know, the stockpile really should have had billions of masks. And I believe it had about 12 million masks left in it. I mean, like nothing. Because people are, you know, and when you're in a hospital working in a crisis, like you're putting masks on and then you're like disposing of them like right away. So you go through lots of masks, each healthcare worker. So that's a really easy thing. It's incredibly easy, and we didn't do it. So there are a bunch of places where we need to do a lot better.
Good morning, Mrs. Zimmer. My question for you regarding the COVID pandemic and your reporting on it as a whole is that, what is your opinion on the credibility of science news, especially after the COVID-19 pandemic?
I think that, you know, I think that some science writers like did their very best work of their whole careers during the pandemic. You know, this is something where we were trying to report on something, basically like trying to like keep up with the scientists who were learning things for the first time. You know, it's not like all the answers were there at the start of the pandemic. People didn't, you know, we didn't know how this virus spread, for example. We didn't know what immunity to this would look like. We didn't know whether vaccines would work at all. We didn't know any of that. So we were reporting this stuff as fast as we could. When the Pulitzer Prizes were being handed out, they went to people like Ed Young, for example, at the Atlantic, and others specifically for covering the pandemic. So I think a lot of great work was done. That being said, you know, I think there's a lot of, I think that, you know, I think journalists sometimes like were making, you know, there definitely were mistakes that journalists made as new stuff was coming out. So I'll give one example. So Omicron shows up November 2021. And I remember writing about that for the New York Times. Very, you know, the news breaks, got to get a story out. And I noticed that other journalists, when they would talk to me about it, they would seize on the fact that, if you looked at the first cases coming out of South Africa that were in South Africa, recorded cases, a lower percentage of the people who got Omicron went to the hospital than earlier strains. So people would say, "Oh, it's milder," and I would say, like, that's no, that word, don't use that word. But then people use that word. And, I think maybe at every stage of the pandemic, people want to say, like, oh, it's over now, we're done. And that's just not, the virus doesn't care. It just doesn't care what we want. It's just going to do its thing. So the reason that Omicron was sending a lower percentage of people to the hospital was because, first of all, it was because a lot of people in South Africa had actually already gotten infected or had been vaccinated already, and so that actually provided a sort of a protection against really severe disease. It was not so good at providing protection against infection, just having the virus grow inside of you. And that's because this virus had evolved to evade antibodies from either a previous infection or from vaccination. It still, you still did have some protection, but just not as much as with the earlier forms. So you put that together, and what that means is that you have a variant that infects way more people than anything in the pandemic before. So if you look at a graph of cases, you will see that in December 21 and January 2022, like, it shot up, and people who hadn't gotten sick till then, start getting sick. I was one of them. Like in January, I got Omicron, and it wasn't great. I wouldn't recommend it, but I mean, I didn't go to the hospital, that's for sure. And I credit having been vaccinated for providing me that kind of protection. In the same way, like a flu vaccine will work. But the fact is that when you get so many people, such a huge number of people, even if it's a smaller percentage, the absolute number of people is bigger. So hospitals were slammed. We had a huge spike in deaths. So from a public health perspective, from looking at what's our health like as a group, it was terrible. It was not mild at all. And that was, I think journalists could have done better at making that clear. It's a tricky kind of point, but it's an important one.
Hi, Mr. Zimmer. I read one of your actually more recent articles of the ancient DNA revealing the history of hunter-gatherers in Europe. And I just had a couple questions on that one. Do you prefer investigating like strictly scientific studies and discoveries, or as in this article, like, do you enjoy when science and like history or literature interact?
The nice thing about this job is that you don't have to like choose one over the other. Like, so some days I'm writing about science that, had some important intersection with history. Sometimes, you're just writing about some weird animal that lived 200 million years ago and has nothing to do with human history. Science is present in lots of different kinds of news. And so I like to I like that variety.
And more specifically about this article, what would you say was the most massive difference in genetic structure of the DNA between modern day Homo sapiens and the discovered past populations?
There's not like, when you talk about these sort of, genetic differences, like, it's not like one thing. I mean, when geneticists talk about this, what they're saying is, like, if we look across all of your DNA and compare it to someone else, someone else's DNA, do you have lots and lots of mutations that, distinguish the two of you. And sometimes you can actually see people, people with ancestry in different parts of the world. You can start to sort of make out some of those genetic differences. But, the fact is that, if you add up those genetic differences between, say, let's say, just trying to think of an example, let's say you're someone who is an Inuit in Alaska and someone who is descended from, people in Angola in Africa. Let's say that they both get their DNA sequences and they say, okay, let's compare it. They will find, like a million differences between their DNA, but that's like less than 1% of their DNA. It's less than 0.1% of their DNA. So you can find the mutations that allow you to distinguish different groups of people, but that does not mean that they're somehow like profoundly, fundamentally different. So what you can see when you look at Europe is that, when Homo sapiens showed up in Europe, they probably showed up several times. And each group was very distantly related to the other groups. And it seems like for a long time, people would show up and then just disappear. And they just didn't, maybe they lived there for thousands of years and then they just vanished. And then it wasn't until about 40,000 years ago or so that at least a couple different groups migrated into Europe, and they are, their descendants became part of the rest of European history.
Hey, Mr. Zimmer, I also read that same article, and I was wondering how DNA is extracted from ancient bones and teeth.
It's a really cool thing. I mean, it's kind of weird because when I started as a science writer, this was literally like science fiction, like, Jurassic Park had come out, this novel, and then the movie comes out and people are like, yeah, that good, good story, but this would never happen. And then it turned out that, you could actually take, a chunk of bone, and grind it down. And then you'd apply a series of chemicals to it that would pull, would pull away the bone, would pull away, any organic molecules inside except for DNA, and then would fish out all that the DNA, what DNA was in there. The problem is that in a lot of cases, what's in there is not, let's say you're taking a Neanderthal jaw. The DNA that you're going to get may actually be from bacteria that have been living on that bone for 50,000 years and just sort of growing, and just because it's in the dirt. So so scientists will be super careful, and they will sort of drill in deep into bone to places where bacteria can't get to. But there are other problems, some of these things have been handled and they're sitting in museums. So now they've got human DNA on them, the people who are holding them. So how are you going to tell the difference between the Neanderthal DNA and the human DNA? And one of the things you can do with ancient DNA is actually that over thousands of years, the DNA falls apart into fragments. And then sometimes a little bit of radiation will cause a little bit of damage to those fragments. And you can actually scientists have learned to recognize fragments of DNA that look old. It's like looking at somebody and saying, there's an old person. You can look at DNA and say, that's old DNA. And then you read those sequences, and then from that, you can start to figure out what the sequence of genes are in, say, a Neanderthal.
So I read your article
she has her mother's laugh, and I was wondering, in genetic testing, how do scientists trace an individual's DNA back hundreds of years to find their ancestors?
Well, if in the book I talk about sort of I talk a bit about how these companies like 23andMe or Ancestry will try to connect you to relatives with your DNA. And the simplest example would be like, let's say that you and a sibling got your DNA sequenced. One way you could recognize that you were really closely related because you would share a lot of totally identical stretches of DNA. And that just has to do with, the fact that, your parents, in that process of producing you, their chromosomes, they're taking one copy of each pair of chromosomes, and then there's this process of shuffling the DNA through meiosis and then combining them to make your genome. And so, roughly maybe 50% or, some figure like that of you can see if you look at the DNA carefully, you have identical stretches with your sibling. That's just the statistics of it. And so you can be confident about that. This is, a brother or a father or, some really close relative. If you go, if you're going to do a first cousin, well, what do you share in common with that cousin? Well, you share grandparents in common. So from that original DNA from your grandparents, each of you, one of your parents was born, but then there was somebody else who married your parents. And so that brought in different DNA. So now those stretches of identical DNA, they're shorter and they're fewer. So so if you look at that and you're like, if you look at the percentage and you can say, oh, that looks like a first cousin. There's less for a second cousin and so on. Now, you go back a few centuries and it starts to get really hard to use that method anymore because those identical stretches are so rare that it's hard to be sure what kind of relationship you're looking at. But that's the basic method, and it's good enough that people will discover all sorts of things on these sites. They will discover lots of relatives they didn't know they had. Sometimes people discover things that they might not have wanted to know. Like maybe a parent is not actually their parent, or a grandparent is not actually their grandparent biologically. Those are the kinds of things that we can now learn by looking at our DNA.
I read another article of yours. The oldest known DNA offers a glimpse of a once lush Arctic. And I was just wondering, kind of surrounding some topics in there, but what's the current state of de-extinction? Would there be a way to reintroduce some of these earlier species from previously collected DNA sometime in the future?
I wrote about de-extinction, actually, a cover story for National Geographic about, like 10 years ago now. And, the idea is very appealing, but actually trying to do it in practice is pretty hard. And so no one's really done this yet in any serious way. But there is actually a company now called Colossal that has a business model that they are going to start making mammoths, or at least elephants with sort of mammoth-like characteristics, in the next few years. And they want to make dodos and they want to do other things. We humans are responsible for a lot of species that have become extinct, like mammoths and dodos. And we are currently driving a lot more species extinct by cutting down forests, and with pollution, and increasingly with climate change. So, people have made the argument that like this is part of our obligation to nature. But if you make a mammoth, what do you do with it? Where do you put it? You could have 3 million years ago, you could have stuck it in Greenland and it would have had a great time. But Today, we haven't just driven the mammoth extinct. We have changed the habitat. So there aren't that many places on Earth where it could work. There are some, and some people have said that there are places in, say, Siberia or Canada where you could put them back. But I'm kind of skeptical.
So, hey, real quick, if you could, who would you bring back first?
Oh, what's it called? The stellar sea cow. It's
okay.
If you know, like manatees or dugongs. So there was this thing in the North
Yeah, we got
Pacific.
manatees here.
Yeah. So imagine something that's like, I don't know, I think about 4 or 5 times bigger, like that kind of thing, but 4 or 5 times bigger, swimming around in the North Pacific. They were there until the 1700s, and they were just hunted to death by, particularly by Russian, hunters who were, whaling and killing other animals for oil and skin and so on, just for the commercial market. So they were there not very long ago, and they just, if you look at pictures of paintings of them, they're bizarre, they're so weird. So that's my personal favorite.
All right, and just we probably got time for 2 more questions, I think, something like that.
Hi, Mr. Zimmer. I read your article that was talking about how science museums had taken stock of over 1 billion objects from around the world, and I noticed that hundreds of millions of those objects were insects. And my question was what you thought on the extent that insects have shaped like speciation through like carrying disease and like pollination in the world.
So yeah, I mean, we tend to not think that much about insects in our day to day, but insects are, the by far the most, the biggest number of animal species are insects. They just, we don't even have we're so far from even knowing all the species of insects on Earth. There are just so many left to be discovered, and they're hugely important. And so when we lose an insect species, there are ecological consequences. And and there are all sorts of opportunities to just to learn about how nature works by looking at insects. They're just amazing, just in their own way. And so museums do have a lot of insects, but when you think about all the insects on Earth, they actually could use a lot more. They're not, museums are, if museums are trying to sort of capture a snapshot of the diversity of Earth today, they need a lot more insects.
Hey, Mr. Zimmer. So this will be our last question. And I was reading your article titled "Scientists Are Designing Artificial Proteins for Your Body." And my question was, what are some of the ethical issues surrounding scientifically designed proteins for humans?
Yeah, good question. I mean, it's I mean, a lot of medicine that we take, medicines for, to cure cancer, for example, are just designer proteins. They're proteins that someone like found, and they're like, okay, this is going to work to save somebody's life. But if you know about like nerve gas or botulinum toxin, poisons, those could be proteins too. And if you've got like GPT looking around, just trying to like think of new proteins, if it's given directions to find proteins that might cure cancer, it may very well find them. And if you give it instructions to find, the next nerve gas, it might find those too. So there aren't a lot of regulations. There need to be.
Awesome. Well, hey, that wraps it up. Carl, thanks so much. This was awesome. Really appreciate your time. All right.
Yeah,
we'll be in touch. Yeah, I'll send you a copy.
All right.
Thanks so much.
All right. Bye-bye.